We'll continue talking about enzyme catalysis, and here on this slide you see a list for 4 catalytic mechanisms that we need to study in this course: RNase A, serine proteases, and carbonic anhydrases. These 3 mechanisms we already covered in this course, and today we are going to talk about the catalytic mechanism for lysozyme. Lysozyme is an enzyme that destroys bacteria cell wall, and by doing that it deprives bacteria cell wall from structural integrity, and bacteria dies. So you can say that lysozyme is a part of innate immune system in animals, and it is found in such liquids as tears, saliva, human milk, and mucus. On this slide, you can see structure of typical bacteria cell wall, which consists of peptidoglycans, and this specific peptidoglycan consists of 2 alternating residues of N-acetylglucosamine, the structure that you already learned in this course, and N-acetylmuramic acid. It's a new structure, but the difference with N-acetylglucosamine is minor. It's at Z position, so you can see a lactic acid residue attached here, and this lactic acid residue serves as a point of attachment for a peptide that actually cross-links the long polymeric polysaccharide chains. Okay, lysozyme breaks the glycosidic bond between N-acetylmuramic acid and N-acetylglucosamine. So this glycosidic bond is hydrolyzed by the enzyme, and the reaction is shown here. Actually, the second reactant is water. Don't forget water as a reactant. And again, this N-acetylmuramic acid. There is no peptide here shown, just lactic acid shown, and the glycosidic bond, this glycosidic bond is hydrolyzed. Okay, so you can say lysozyme is a hydrolase according to classification of the enzymes. So just taking you back to non-enzymatic acid catalyzed hydrolysis, the mechanism that we studied in module 1. So this is the mechanism, how glycosidic bond in polysaccharides would be hydrolyzed otherwise. And just to mention that this reaction requires harsh conditions. It's concentrated HCl at 70 centigrades several hours. And a lysozyme, shown here, facilitates the rate of uncatalyzed reaction by 10 to the 8 times, so which is very impressive improvement in catalytic rate. Now it's a small enzyme, 14.3 kilodaltons only, 129 amino acids. The 3-dimensional structure in complex with its substrate reveals a cleft in which 6 units of polysaccharide are bound. They are indicated by letters A, B, C, D, E, and F. And you can also see here catalytically important glutamic acid 35 and aspartic acid 52 next to the cleavage site between rings D and E. Okay. When we talk about catalytic mechanism of lysozyme, we will cover again general acid-base catalysis, covalent catalysis, proximity and orientation effect, and preferential binding to the transition state complex. So on this slide you can see another representation of catalytic site in lysozyme. Now you can see all in chemical details the substrate. Zoomed in this picture zooms us to rings D and E, and you can see cleavage site, the hydrolysis site, and some catalytically important amino acids that bind the substrate and also catalyze the cleavage. And again, I would like to focus on aspartic acid 52 and glutamic acid 35. These are the two amino acids that we are going to consider in details. The other two things that I would like to mention is the deviation from chair conformation in residue D. So this ring is not in chair conformation as it's supposed to be, as ring C and E are. It's in half chair conformation, meaning that these three atoms, oxygen, carbon number 1 and carbon number 2, make a plane of the molecule, and the same in the same plane with carbon number 5 and 3 right here. Okay, and the other detail is that this ring, D ring, is an N-acetylmuramic acid ring, which is indicated by R, which stays for lactic acid residue. And this R cannot be accommodated neither in C binding site or E binding site. That's how enzyme cleaves only after N-acetylmuramic acid, because this R can be accommodated in the site that corresponds to the binding of D ring. At the first stage of catalytic mechanism, we have protonation of oxygen, this bridging oxygen, and this is the same stage as it is found in just acid catalyzed hydrolysis mechanism. But now this part becomes a good leaving group after protonation, and it is released from the enzyme center. So now, why is it released so quickly and efficiently? It is because the resulting carbocation is stabilized by negative charge of aspartic acid right here. Moreover, as I mentioned before, just upon binding, the half-chair conformation already prepares this bond for the cleavage. The empty orbital of carbon number 1 with a positive charge is now conjugated with a lone electron pair of oxygen, which is also reflected by this resonance form, this double bond, CO double bond. This stabilizes the carbocation, and this oxonium ion stage is very important for overall catalysis. And now we have attack of aspartic acid, negatively charged aspartic acid on this group that forms a covalent adduct, so covalent intermediate. So this covalent intermediate is stable compound, and it can be found, it can be isolated, it can be characterized, and you can say this is minimum in an energetic curve. And then water enters the active site, and on the next stage, glutamic acid 35 acts as a general base to remove proton from water molecule and increase its nucleophilicity, and we have hydrolysis of this CO bond right here between aspartic acid and the sugar unit. And the second part of polysaccharide is released in solution. Let's walk again through the mechanism of lysozyme catalysis and identify catalytic mechanisms that the enzyme uses to cleave efficiently the glycosidic bond. First to note is preferential binding of the transition state. You can see ring D is in half-chair conformation. Indeed, this is a crucial step to stabilize the half-chair conformation structure, which is oxonium ion. Since the formation of oxonium ion is the limiting stage of this reaction. The other strategy that we can identify here is general acid catalysis accomplished by glutamic acid residue. And next we have covalent catalysis by aspartic acid, formation of covalent stable intermediate. And the other type of catalysis is general base catalysis accomplished by deprotonating glutamic acid residue, which pulls away proton from water. At the end of this part of our course, I would like to remind you again the classification of enzymes and give you at least one example for each of the type of the enzymes. For example, oxidoreductases, catalyzed redox reaction, and a couple of them I mentioned in last lecture. One is alcohol dehydrogenase that use NAD plus to oxidize alcohols. The other one is cytochrome P450 that oxidizes a bunch of different compounds using oxygen. Transferases, some of them we already learned. For one transferase is DNA polymerase. It's a nucleotidyl transferase. Other transferases that are going to be mentioned in this class are protein kinases and enzymes that transfer a gamma phosphate group from ATP molecule on proteins. We studied many hydrolases. One of them is lysozyme that I just mentioned. Others are serine proteases, RNase A, and other protease is myosin and actin. Those that I mentioned when we studied muscle contraction. Lyases. One lyase that will appear in this course is adenylate cyclase. It forms a bond, a new bond, using ATP as a starting substrate, and it synthesizes cyclic AMP out of ATP and releases pyrophosphate. Isomerases, proline racemase is an isomerase that was mentioned in the last lecture, and ligases. Out of ligases, we only mentioned DNA ligase that is used in molecular cloning to seal two strands of DNA. Please remember at least one representative for each class of the enzymes.